Welcome everyone to my poster. Thanks for attending. Today I'm going to talk about how we can use multiparametric flow cytometry to understand better the human cardiomyocyte cell cycle. Congenital heart diseases are a leading cause of deaths among infants. Several congenital heart diseases, including hypoplastic left heart syndrome, are characterized by altered cardiac tissue architecture and the loss of cardiomyocytes, with some of these associating with increased DNA damage and abnormal myocyte proliferation. However, studying the human cardiomyocyte cell cycle is challenging, is complex, and therefore its behavior and progression remain elusive. Ultimately, the challenge is to combine several cell cycle markers to understand better and map its complex progression. Here is where flow cytometry fills the gap. Human-induced pluripotent stem cells offer an unlimited resource to study the human cardiomyocyte cell cycle through flow cytometry. Why? Because we can determine at the single cell level, using different palettes of colors or fluorophores, with highly accuracy, the cell cycle progression of these cardiomyocytes. In our study, we first compared the efficiency of different broadly used cardiac dissociation strategies to obtain single and viable cardiomyocytes via flow cytometry analysis, applying them to 2D or 3D iPSC-derived cardiomyogenic cultures. Our results, as you can observe here in this plot, show that prepained excels in generating single viable cardiomyocytes, as evaluated by the exclusion of propionate iodide and the incorporation of calcium AM into these cardiomyogenic cultures. Next, with this in hand, we explore the cardiomyocyte cell cycle progression at day 12 of cardiogenic differentiation, and for this aim we use DNA ploidy, Ki67 EDU, and phosphohistone 3 staining coupled to cell trace to understand the human cardiomyocyte cell cycle and division using multiparametric flow cytometry. Because of time, I'm only going to show DNA fluorescence and Ki67 labeling, as you can observe here. After this, a gating strategy to determine single cells. As you can observe, a large majority of cardiomyocytes in this plot in the middle are in G0, G1 phases, even though a significant fraction of them is positive for Ki67, and in this case, 20% of them are positive using this in-house media. As I mentioned, we tried two different differentiation protocols, obtaining different proportion of Ki67 cardiomyocytes, and an in-house developed protocol yields higher Ki67 cell cycle competence cells compared to the commonly used stem cell differentiation protocol. Using Ki67 mean fluorescence, we next determined that the quantity of Ki67 per cell is higher in G2M or 4N cardiomyocytes. Thus, our data highlights the accuracy and high throughput potential of flow cytometry to map the human cardiomyocyte cell cycle and provide a better entry point for deeper analysis of cardiac cell cycle behavior in homeostasis and disease. To finish, our current experiments aim to unveil further details into the human cardiomyocyte cell cycle using high-dimensional flow cytometry of fluorescence analysis at the single cell level, and also to examine patient-specific disease modeling of hypoplastic left heart syndrome or other congenital heart disorders using 3D cardiac organoids or engineered micro tissues or micro hearts. Thanks again for attending to my posters, and if you're interested, please get in touch.